Hello everyone, my name is Dita O'Boyle and today I'm going to be talking about Water Quality Parameters 101. Now I work at Rookery Bay National Estuarine Research Reserve and we are part of a nationwide system of research reserves. There's 30 of us. We're all located where there are estuaries. An estuary is a place for two different types of water mix. That is usually salt water from an ocean and fresh water coming from over the land. So with Water Quality 101, we're going to be learning about what parameters scientists look at to see if the water is healthy. But first of all, what kind of things do we do to see if we're healthy? Well, often we go to a doctor's office, and depending on the type of checkup, we may get our temperature checked, look at our reflexes where they hit your knee, look at your weight, height, see how much you've grown, check your blood pressure, look at your lungs. Lots of different measurements depending on what you're getting checked on. Now, I don't think you can check the water's blood pressure, but there are a different series of measurements you can look at. Now, since we are part of this national network, each one of these reserves has the same set of parameters that they look at to see if the water in their area is healthy. And we're going to be walking through those today. So we're going to look at what depth is, the temperature, dissolved oxygen, salinity, pH, turbidity, and chlorophyll. And all these different measurements or parameters all interact with each other. And since we're looking at estuaries, these are really crazy places because these parameters are constantly changing because it's that mix of fresh and salt water. So we're looking at the depth of the water. This is fairly important because this changes regularly. In our oceans and estuaries, we have tides. There are 4 tides a day, 2 high and 2 low, and they're caused by the moon's gravitational pull. How big of tide you have depends on the cycle of the moon and then also the time of year. So here's an example of some tidal data from Rookery Bay Reserve. This is looking at one day, so from the morning on March 10th to the end of the day. And you can see that at its highest point, it's at about 1.6 meters. A meter is roughly 3 feet. And at its lowest low tide, it gets down to about 0.6 meters. Now that doesn't seem like too much of a change, but if you were a manatee trying to swim in that area, probably couldn't go in that part of the estuary when it's down at 0.6 of a meter. Now our next measurement is temperature, and this does tie into that water depth because if your water is shallower, it's going to heat up quicker by the sun's rays. And when we are looking at our temperature, we can use a few different tools. We have a regular thermometer here and then also a digital probe that's looking at the temperature. And a lot of times when we're checking the temperature in our house to see how hot it is outside, if we wear a sweater or a t-shirt, we're going to look at Fahrenheit. But since we are collecting scientific data that we want to share with other countries, we are going to use degrees Celsius. Now this is kind of a crazy graph, but this is looking at the temperature in the reserve in one of our bays over a whole year. So starting March 2019 all the way up to March 2020. And you can see that it fluctuates quite a bit depending on the time of year. Now, since we may not all be familiar with calculating Celsius from Fahrenheit, we can look at, let's say, right here looks like it's one of the hottest points in that year. It's in July, which makes sense, and it's about 35 degrees Celsius. There's an equation at the bottom that will show you what that is in Fahrenheit. And this temperature is a pretty big factor as well, because this temperature is going to affect our next parameter, which is dissolved oxygen. And there are some animals out there that really like cold water. Maybe you're only going to find them up in the Arctic Circle. There's some that like slightly cooler water, which would be our temperate animals. There's animals that only like really hot, warm water. Those are going to be our tropical animals. And Southwest Florida is a pretty cool place because we are subtropical. So we get kind of a mixture of animals. We get some of those more northern animals and also the more tropical animals. Hopefully that gave you a little time to do that equation at the bottom, but your answer is 95 degrees Fahrenheit is 35 degrees Celsius, which sounds about right in July here in Southwest Florida. So as I mentioned, temperature does affect dissolved oxygen, our next parameter. So we, mammals and other animals on land, we breathe oxygen. We take it out of the air with our lungs, but the animals that live in the water, such as fish and invertebrates like crabs, use their gills to take out dissolved oxygen. And just like how maybe at a higher altitude, if you're up at on a mountain, it's harder to breathe because there's not as much oxygen in the air. Sometimes there's water that doesn't have much oxygen. And when we measure oxygen in the water, we can either use milligrams per liter or as a percentage. So this little kit over here is a dissolved oxygen kit. You can either use a fancy probe to test dissolved oxygen, or you can do this color test. And how it works, Morgan's going to demonstrate in this video. But this vial she's holding has a chemical in it, and when it mixes with the water, it's going to turn blue. And depending on how blue it turns is going to tell you how much oxygen is in the water. Now, this isn't a perfect test because all of our eyes are shaped a little different. They work a little differently, and if you're in the shade or sun is also going to affect what reading you get. But generally speaking, in our estuary, anywhere between 3 and 8 is perfect for our animals here. That being said, since our estuaries are kind of a stressful place because there's so many different things that can change in the water quality, some animals have adapted to very low levels of dissolved oxygen. If you have a creek and there's a little bay back in the mangroves, and let's say you don't really have a big tide for a few days, maybe that bay isn't going to be getting flushed and new water coming in. And if it's in the, let's say, towards the end of the dry season, it might be kind of hot, and that water is going to be heating up. And as that water heats up, the amount of oxygen it can hold decreases. And so then you're probably not going to have as much variety or as high a number of fish in there. But fish like in this picture, this tarpon, can actually gulp air out of the atmosphere. So it doesn't need to just use its gills. Its swim bladder actually is modified to act a little bit like a lung. So it can survive perfectly happy in one of those bays with really low dissolved oxygen. Our next measurement is salinity, and salinity is looking at how salty the water is. And this is a really big one because a lot of animals are adapted to only live in fresh or salt water. Their bodies can't handle the stress of being in a liquid that has a different salt concentration than their body does. But again, our estuaries, these animals are adapted for these changes which happen every tidal cycle. So when the ocean water comes in at a high tide, it's going to increase the salinity, and when the tide goes out, the water is going to become less salty. This also changes depending on the time of year. Southwest Florida, we generally have 2 seasons, our wet and our dry season. So in the summertime, when we're getting all those big rainstorms, our water gets a lot more fresh. In this video, Morgan's demonstrating a refractometer. And how this works is she puts a little bit of water on it, closes the cover, and she's going to be looking for where a blue and white line mix. So this uses light, and depending on how much salt is in the water, the light is going to bend a different amount, and she's going to get a different reading. Now, how she's going to read it is in parts per thousand. So this is looking at how much salt's in the water using a kind of 1,000 ratio. So if you have a salinity of 5, which would be pretty much fresh water, you would have 5 cups of water in 1,000 cups of salt. If you're looking at ocean salinity, which is more at 35 parts per thousand, that would be 35 cups of salt in 1,000 cups of water. The water in our estuary or brackish water, which is pretty much anywhere between like 10 to 27, 28 parts per thousand. Our next measurement is pH. So this one is looking at how acidic or how basic or alkaline the water is. So it's on a scale from 0 to 14, and a really, really strong acid, like the one on the bottom left, sulfuric acid, is gonna have a pH of around 2.75, and this could burn your skin, definitely put holes in your clothing, something you wanna be really, really careful about. But you could have a more mild acid like in orange juice, which only has a pH of around 4.2, and we drink that for breakfast all the time. So a little bit of acid isn't a bad thing, but let's think of the role of acid. So in our bodies, we do have stomach acid and it breaks things down. So if you have water that's really acidic, it actually will interfere with animals that make shells like clams or oysters or crabs. It interferes with the production of their shell or also damages their shell. At the same time, so if a high acid is 0, a really high base can be dangerous for the water as well. So strong base would be like Clorox or bleach. That could have a pH of around 11. But again, a little bit of a base is a good thing, and that could be like in your pool when you add chlorine. That helps keep the water clean and prevent algae or bacteria from growing in the water. So your pools are just at about like 7.2 pH, or maybe up to 8. So if we have a scale of 0 to 14, where do we think we want the water to be at? Well, 7 is considered neutral. But generally in our estuaries, a pH range from 6 to 8 is perfectly fine. A lot of times our pH does run a little bit towards the acidic side, and that's because of all the mangrove trees we have in our estuaries. When those leaves fall off, they release tannic acid, which does lower the pH a little bit. In this video, Morgan is going to be demonstrating how litmus paper works. So it's treated with a chemical, and when you dip it in the water, it's going to change color depending on the pH of the water. So if you get warmer colors, red, yellows, oranges, it's going to be more acidic. And if you get a green or a blue, it's going to be more basic. Our next parameter is turbidity, and turbidity looks at how much stuff is floating in the water. So this could be plankton, it could be sediment. This does not refer to the coloration. And this is a really tricky parameter on its own. It's because it can be affected by what's going on in the water. If you have a lot of waves going on, that's going to be stirring things up and increasing the turbidity. If you have a manatee or a boat swim by, that's going to stir up the turbidity. So it really depends on what's going on in the area right at the moment when you're taking your sample. One way you can test turbidity is using this tool that Morgan has in this picture called a sucky disc. And how it works is that it's black and white, and as you lower it into the water, you keep lowering it until you can no longer see the difference between those two colors, and that's going to give you a rough estimate of your turbidity. Now, in our estuaries, a lot of times it's so shallow that these discs will hit the bottom before we get a reading. And as you can see, the color of the water is kind of brown. And that's because, again, of all those mangrove leaves falling in the water, releasing that tannic acid, which does change the color. And remember, turbidity isn't looking at the color, it's looking at the stuff floating in the water. And this turbidity does tie into our previous parameters, because if you have more stuff floating in the water, it's going to absorb more sunlight, which will heat up the water, which then decreases the amount of oxygen. Turbidity does tie in very closely to chlorophyll, which is the last parameter that all the National Estuarine Research Reserves look at. And chlorophyll are those little teeny cells inside plants. It's pretty much the little power plants inside their bodies that help them produce their food. So this is what absorbs the sun energy and actually makes the sugars that they use to grow. And that process is called photosynthesis. So our land plants have chlorophyll, but so do our algae in the estuaries. And so when you get a really high turbidity level, it could be because you have a lot of algae in the water. And these algae are really important because through photosynthesis, they actually make more oxygen than all the rainforests on land. And so that's huge for the animals in the water to breathe, and also that gets released into the atmosphere as well. So this is another way that the dissolved oxygen level is increasing in our estuary. Now, to measure chlorophyll, you have to get a water sample, and then you actually filter out as much of the plankton as you can using some sort of filtration device. This one uses a hand vacuum pump, but you can do it electronically as well. And then it gets put into a couple different machines to concentrate the chlorophyll, and then also to read how much chlorophyll is in the water. Now, all those measurements we talked about take a while to do if you were doing them kind of the old school methods that were in this presentation. Luckily for our water quality specialist, she uses this handy dandy data son that collects this data automatically, and also some other parameters as well. Has a series of probes. It sits in the water and it's going to collect that data, and it actually gets sent up into a database. So if you have an interest in looking at what's going on in the water in our area, or maybe you're curious about what's going on in Alaska or California, you can go to this website and click on it and get the data from any of the National Estuarine Research Reserves. It'll link you to this page, and the website for this one directly is right here on the left. The previous link has a little bit more information on how to use the data. And what you'll do is you can pick any of the reserves marked by the little bunches of grass. That'll be choosing your reserve. And then at that location, there'll be different water quality stations you can pick. And then you can choose what kind of data you want. Do you want to look at depth? Do you want to look at salinity, the dissolved oxygen? And then you can get that data for any date that's already been uploaded. So it's a pretty amazing, awesome set of data that anybody can use. And there'll be an activity later that corresponds with this data. With that, thank you so much for listening, and have a great day.